Hello guys and welcome back to Excel Academy. So we were discussing about the plasma membrane phospholipids, right? And like how in yesterday's lecture we discussed about the like what is the composition of the outer and the inner leaflet of a plasma membrane, right? So this is the same diagram which we were discussing. So we saw over here that on the outer layer we see only the protruding integral proteins, and wherever here we see both integral as well as the peripheral proteins, right? Now there are certain other types of protein known as lipid anchor proteins. So there are also lipid link proteins. इससे समझ में आ जा रहा है that they are located on the surface of the cell membrane, right? And these proteins are covalently attached to lipids. इसका मतलब क्या? You have got the plasma membrane over here, and to these phospholipids, okay, यहाँ पे जो present है, to these phospholipids the proteins are attached. Okay, so those are your lipid anchor proteins. So these proteins are embedded within the cell membrane. They are linked to certain fatty acids like myristate and palmitate. So guys, this is something which you need to remember. Okay, and these anchors the protein to the cell membrane. This protein to the cell membrane. They play a very important role in protein function. किस तरह का? So mainly in cell signaling, we'll see what or different kinds of peripheral proteins are playing role. ओके तो आपको हमेशा ये याद रखना है, जब भी हम कोई फैटी एसिड लिंकेज की बात करेंगे बिटवीन द लिपिड एंड अ प्रोटीन, इट इज ऑलवेज अ मिरिस्टिक एसिड और अ पामिटिक एसिड. रेयर केस होगा जहां पे आपको स्टेरिक एसिड का नाम आएगा. ठीक है, सो मेनली इट इज मिरिस्टिक एंड द पामिटिक एसिड. Now look at this diagram now. So this is nothing but the same thing, but here with the chemical linkages shown over here. Okay, so see, you have got the plasma membrane. ये हो गया आपका exoplasmic phase. This is your cytosolic phase, right? Now over here, see the amide linkage. What is amide linkage? Wherever CONH bond is there, your peptide bond is also a amide linkage, right? Now you can see here the lipids are there, protein is there, then it is attached. Fatty acylated, this is acylated protein. यहाँ पे क्या है? Myristic acid हमने यूज किया. कहाँ पे आपका myristic acid? Over here. यहाँ पे ही आपका acid, fatty acid can be present. Why? Because it is also a hydrophobic molecule, right? So यहाँ पे आपका myristic acid रहेगा, and that myristic acid is making a amide linkage with your protein. ठीक है, so ये आपका protein हो गया and glycine से attach है. Can you see? This is fatty acylated. Okay, is it clear? The amide linkage is there. Now we have got another examples where we see that if it is a palmitic acid, okay, palmitic acid अगर है तो वहां पे आपका बनेगा thioester linkage. So this is your palmitic acid. It is forming a thioester linkage with the protein. अब प्रोटीन के कौन से अमीनो एसिड? थायोएस्टर है, तो यहाँ पे आपका सल्फाइडल ग्रुप प्रेजेंट होना जरूरी है. एंड दिस ग्रुप इज, यस
Ma'am, the last 30 seconds or so was cut. I couldn't hear anything.
Like for everyone?
No.
It was audible to others, right? I'll just repeat once again, no problem. Okay. Yeah. So I was explaining that, so we have got lipid anchor proteins, correct? Simple bhasha mein, the proteins are attached to the lipids. And though what are the examples? So as I mentioned over here, you have got the myristic acid and you have got the palmitic acid. Okay. Now here we are showing the diagrammatic representation of what we just said right now. So you have got here the outer leaflet and the inner leaflet. This much is clear. Now, if a fatty acylated protein, okay, this is fatty acylated protein is there. If myristic acid is used in order to link the protein, correct, then it is a amide linkage. Okay, so here it is your myristic acid which is forming amide linkage to the protein, and this is formed between the fatty acid ka last COOH and the glycine ka NH group. So we know that also that when a peptide bond is formed, it is again the formation between the COOH of one amino acid and NH of another amino acid, right? So water removal के साथ, condensation के साथ, you get a CONH bond. That is your peptide bond, correct? Here it is amide bond because it is formed between a myristic acid and a protein. Is this much clear now, Shruti?
Yes, ma'am.
Yeah. So what you need to remember here for your exam point of view is that if myristic acid is used in order to link proteins, it is forming a amide linkage between the COOH acid है, right? Myristic acid. तो last में जो tail होगा, last में आपका COOH ही होगा. So this COOH and the NH group of glycine, they are forming the amide linkage. ठीक है? तो आपको क्या याद रखना है? Myristic acid के साथ glycine forms the amide linkage. Okay, done. Now coming to the next. So in the next, what we see, we have got palmitic acid. Again, palmitic acid is having here, ये आपका DO है, okay? Palmitic acid is coming and it is forming a thioester linkage. Thioester means you need to have a self-hydral group, okay? This is your self-hydral group. So self-hydral group कौन से amino acid में present है? There is only one amino acid which has got the SH group and that is your cysteine. ठीक है? So it is going to form a thioester linkage between the palmitic acid, this one, and the SH group of your cysteine. ठीक है? So fatty acylated protein with the palmitic acid. This much clear? This is first type and this is another type, is there? Clear? Then we also have another category where it is farnesyl or geranyl group is there. Now what are these farnesyl, geranyl? ये तो आपके 14 कार्बन 16 कार्बन फैटी एसिड हो गए, ठीक है? प्रेनाइलेटेड प्रोटीन, प्रेनाइल और आइसोप्रीन यूनिट 5 कार्बन यूनिट होते हैं. फार्नेसिल विल बी योर 15 कार्बन, geranyl 20 कार्बन होगा. I think so. This will be more clear when we will do the plant physiology. तो उसमें आपको और अच्छे से पता चलेगा, ठीक है? Okay. So these prenylated proteins, so either that will be a farnesyl or geranyl group will be present. To this, it is forming a thioether group. Now this thioether group again with the help of the cysteine of your protein. ठीक है? So ये हो गया आपका third category. So these are the 3 examples of your lipid anchor proteins. Is it clear? This much is clear? Yes?
Yes, ma'am.
Okay. Now what we have another group is that here. See. So you have got here the GPI, glycosyl phosphatidyl inositol. Okay, it is inositol. Here you have got the inositol. ये spelling group, पता नहीं spelling कितना mistake है यहाँ पे, sorry for that. So you have got the inositol. To this your 4 tetracycline has been added. ठीक है? तो GPI का क्या काम है? Again, can you see where is it present? It is present in the exoplasmic phase. Correct? GPI, glycosyl phosphatidyl inositol. It is present in the exoplasmic phase. So इसका काम किसमें होगा? Again coming to the cell signaling. We'll come to that. Okay? जो भी important points हैं उनके हम detail में हम पढ़ने वाले हैं. The structure and where it is having its main function. Okay. So I hope this slide is clear to everyone. Yeah. Okay. Okay. Now this diagram, this table is there. Same thing, it is same thing, just now what we have discussed. So the membrane proteins are covalently bound to the lipid molecules, right? And different categories are there. So ये हमने अभी just पढ़ा the third category वाला, जहाँ पे isoprene groups are used, like for example farnesyl हो गया C15, geranyl C20, okay? And they are attached to the cysteine residue at the C terminal by a thioether linkage, right? Now coming to the fatty acyl group, यहाँ पे myristic acid है, palmitic acid है. Now in the myristic and palmitic acid, what is happening? हमने अभी डिस्कस किया था myristic acid glycine के साथ amide linkage बना रहा था. Palmitic acid is making your anyone? What kind of linkage was formed with palmitic acid? Thioester. Very good. Okay. With again with the cysteine. In the myristic, it was glycine, correct? Now, last is the glycosylated phospholipid, that is your GPI. So, GPI is a derivative of P1 in which inositol have a co-tetrasaccharide, which is covalently blended to the phosphoethanol amine. Okay. Now, it is also attached to the C-terminal amino acid via amide linkage. I have no idea why these many mistakes are there. Okay. So these are the examples of your lipid linked membrane proteins or lipid anchored proteins. Confuse नहीं होना, ये सारी same चीजें हैं, ठीक है? Okay. Now these are the things which we are gonna discuss in detail with each example. Okay, so पहला क्या है यहाँ पे? तो this is just the introduction. अभी भी हमने cell bio properly शुरू नहीं किया है. Okay, we are just at the starting only. Because first we need to understand the plasma membrane structure, then only the rest of the things we can understand. So your plasma membrane and the proteins which are associated are used for the transport. They have got the enzymatic activity, of course, for the signal transduction, cell to cell recognition. So you can see that यहाँ पे दो है, दो cell membrane है यहाँ पे, right? So एक का glycan रहेगा और दूसरे का protein रहेगा, which is going to interact to understand intercellular joining. So where we are going to see such things in the case of the cell adhesion molecules. Okay, so जहाँ पे we will see that these cells are connected together. For example, in the case of your intestinal lumen, so there you are going to have these cells which are connected to each other, right? And they are connected so tightly with each other that the gap between these will not allow even water also to pass through. ठीक है, ऐसा नहीं कि आपकी बॉडी में पानी ज्यादा हुआ तो वो वैसे ही पास कर जाएगा. नो, इट वोंट. For even for water also to pass, a proper proteins are there. I hope you must have heard about the aquaporins. Okay, so this is present in if you want to have water to pass. See, अब भी अगर आप osmosis के बारे में पढ़ोगे, so you know that the water molecules can pass easily. Okay, but when we say osmosis, you need to understand the water movement is totally depend on to the solute concentration. So if you know जहाँ पे आपका high solute concentration होगा and low solute concentration होगा, so in which direction the water will move? Anyone? Tanvi? Yes. Akshaya? I repeat my question. So what I'm asking is, if you have high solute concentration this side and membrane is present and low solute concentration this side, in which direction water will move? From high to low in this direction or from low to high?
Now from higher to lower.
No. When water is there, I'm asking the direction of the water, right? तो जहां पे आपका solute concentration कम होता है, it means there the water concentration is high. जहाँ पे solute concentration less है, वहां पे water concentration is high. Where you have got high concentration of solute, there low concentration of water. So the water movement will be from low solute concentration to high solute concentration. ठीक है, अगर मुझे वाटर के रेस्पेक्ट में बोलना है, देन आई विल से हाई पोटेंशियल ऑफ वाटर फ्रॉम हाई टू लो. देन वहां पे हम कॉन्सेंट्रेशन नहीं बोलते. वी डोंट से वाटर इन टर्म्स ऑफ कॉन्सेंट्रेशन, राइट? बिकॉज वी कैन नॉट कैलकुलेट वाटर एज सल्यूट. सो वी टॉक इन विद रिस्पेक्ट टू वाटर पोटेंशियल. सो फ्रॉम हियर दिस इज गोइंग टू हैव हाई वाटर पोटेंशियल, राइट? ऐसे बनाते हैं, वाटर पोटेंशियल टू लो water potential. Is it clear? I hope you must have studied in your school or I don't know where. So water potential is given with the water potential of solute plus potential of gravity plus potential of pressure. कभी सुना है इसके बारे में? No?
Yes, ma'am.
सुना होगा ना? Yeah, I'm sure because this was very basic when we talk about the movement of water. So I'll just give a brief idea what it is. So water potential is equal to potential of gravity, solute and pressure. So this factor comes when we talk with respect to plants, क्योंकि वहाँ पे हमें turgor pressure and osmotic pressure के बारे में बात करना होता है. Then we consider this. Okay, then solute concentration, you know, जहाँ पे आपका solute concentration ओके हाई है, वहां पे आपका वाटर पोटेंशियल कम होगा. ओके, नाउ व्हाट अबाउट ग्रेविटी? if the potential, if the water movement is along the gravity, it is going to be positive. If it is against the gravity, then it is going to be negative. Now, where do we see the potential gravity going plus and negative? कोई समझा सकता है? Akshaya?
Ma'am, it can be negative when the plants uptake the water, like the vessels take up the water throughout the plant.
Yes, correct. So when they take water from soil, that is roots, and they need to give it to the tip of the leaf on the top, right? So then your water potential is negative and positive जब वो नीचे की तरफ दे रहा है. Okay, simple. So that's how your main water potential is being calculated. Okay, anyways कोई questions तो नहीं आता है. So far I have not seen any question, but it's always good to know. Okay, so I hope this is clear. And the last one is about the attachment to the cytoskeleton and extracellular matrix. So here also we are going to discuss like how the cell membrane and the proteins are interacting with the cytoskeleton. So what are different types of cytoskeleton we know of? So microtubules are there, microfilaments are there, actin, okay, and then we have got the intermediate filaments. So all these three are the cytoskeleton of your cell. Cyto यानी कि cell skeleton, जैसे हमारा skeleton है उसपे base आपके muscles and tissues are present, right? In the same way, the cell also have got the skeleton which is maintaining the entire structure. Okay, so those are your microtubules, microfilaments, and intermediate filaments. We'll talk about this in detail. Okay, so based on this, the entire first topic of your cell biology is based. ठीक है? तो जब तक हम ये खत्म नहीं करते, main topic we won't be able to start. So I'll just give you right now brief idea what all we are going to study in cell biology. So first is all about the plasma membrane, different types of transporters, enzymatic activities, signal transduction. Then different topic will be there, like how each organelles, like starting from the nucleus to the cell wall. So we have got mitochondria, we have got chloroplasts, peroxisomes, and different other organelles are there, right? So about each organelle we need to discuss and how one protein which is synthesized, okay, is being transported to all these organelles. Uska kya mechanism hai? That protein sorting, okay, that will be discussed. Sorting. Then comes your cell signaling. So when we talked about cell signaling, of course, cell to cell we are going to discuss, okay. Then how it is being done, different types are there, and etc. Now this is there. Then there is something else, what the cell recognition. So cell signaling, cell recognition, another here we need to discuss how between a host and a parasite it is happening. So ये भी हमें discuss करना है with respect to insects, with respect to animals, with respect to plants. Okay, so ये थोड़े main important topics है which we are going to cover, and of course about the cancer, basics of the cancer. Okay, okay. Now, yes, the story of malaria and glycophorins. Remember, guys, I was talking about the glycophorin. Do you remember what glycophorin is? हमने कहाँ discuss किया था? Anyone?
It is associated with the RBCs and it is an integral protein.
Yes, and more things with it is absolutely correct. And what else? Main discussion was there about the negative zeta potential. What was that?
Sialic acid which is present, which gives a negative charge to the glycophorin.
Yes.
And the
yeah, so we discussed that glycophorin, it's actually an integral protein, correct? And it is having glycophorin, right? So it's an integral protein which is having the sialic acid as the glycan residues, which is giving the negative charge to the cell surface of the RBC. So because of which it has got the negative zeta potential, and it helps in repelling, that is helping them not to get aggregate or not to form aggregates with the other RBCs, right? And that's how very independently they can flow in the blood vessels. Okay, and that time it is why I told you that it has got a very good relationship with the malaria. Okay, so that is what we are going to discuss now. It is important, it has come in the exam. Okay, so Plasmodium falciparum, so you know that this is the parasite, okay? Invasion ligands and the human receptor. So here what you have to remember, okay? मैं आपको धीरे धीरे बताती हूँ. It is actually very important to remember. So we have got glycophorins from A to E. That means 5 तरह के glycophorin है हमारे पास, okay, in humans. Now this is your reference erythrocyte. Now, can you understand this figure? What is happening? This is your RBC. The concave structure of RBC is there, right? We all know that it has got the concave structure because of the absence of the nucleus and the other organelles. This is your malarial parasite. So what is happening here? The malarial parasite and the RBCs are having a interaction, cell to cell interaction. Can you see? Now that is what is shown over here. So the blue वाला जो है ये आपका malaria का हो गया. Okay. The red one is your human RBC. Now look at that. So the human RBC have got glycophorin A. Do you remember band 3? Right? Ankyrin, spectrin, this all we have discussed. Ankyrin का क्या काम है? Band 3 spectrin को connect करके रखना. So ankyrin is your peripheral proteins. Band 3 is your transmembrane proteins or integral proteins, right? Now here if you see, we have got 4 types of glycophorin: A, B, C, and E. Okay, D they have not shown. Okay, now look carefully. So EBA 140, 175, and EBL1. What are these? These are the receptors of your malaria. Okay, they interact with the glycophorins of RBCs so that they can infect the RBC, go inside and start living there and cause malaria. Okay, are you able to see? Are you able to understand? EBL1 interacts with glycophorin B, EBA 175 with A, EBA 140 with C. This is normally the case, and they invade the RBC using this glycophorin, go inside of the RBC and start growing there. ठीक है, वो eggs जो भी release करते हैं, and they complete their life cycle inside the RBCs. Okay, so so we don't know the ligand about this, so that's okay. This is the normal scenario. Now, if you know, there are people who are resistant to malaria. Why? Because they have got different type of RBCs. Okay, why? Because their RBC have got hybrid of glycophorin, which is having A and B together. Okay, now because of this hybrid, hello, hi everyone, are you able to follow?
Yes, ma'am. Yes, ma'am.
Yeah, okay. मुझे लगा मैं बोलती जा रही हूँ and people are not following. Okay, now if you see the same thing, okay, so these people are, see, your malaria is same, having all the receptors, okay, but this person, okay, this person have got different expression of the proteins on the RBC. So what is the difference? So they express the hybrid of glycophorin A and B on their surface of RBC. Now, because of this, what is happening? Even though the association of this receptor is correct, even this is correct, but here it is not fulfilling the requirement. हमने क्या देखा यहाँ पे 1, 2 और 3 and the person gets malaria. Okay, but here because of this hybrid nature of glycophorin A and B, EBL1 is not able to interact properly with the glycophorin B. Hence, this requirement is not fulfilled, because of which the malaria parasite is not able to invade the RBC of this kind of protein expression on the cell surface of the RBC. Okay, and that version is called your malaria resistant Dantu erythrocyte. Okay, clear? I'm sure, I don't know how many of you must have heard about this, but yeah, if you know that in the blood grouping system also, apart from A, B, O blood grouping, we have got different other methods also. MNS blood grouping method is there, DE is there, बहुत सारे हैं, around 14-15 methods are there. overall in the world ABO system has been accepted. ठीक है? Is this much is clear?
Yes, ma'am.
Yeah. So this is the reason why there are people who are resistant to malaria. उनको कुछ भी हो जाए, the malaria is not going to infect them. Okay. Yeah, and that is all because of the hybrid glycophorin. So that's why I said it's a story about the glycophorin and the malaria. So there are people resistant to the malarial attack. Okay. I hope this is clear. एग्जाम में आपको क्या ध्यान देना, it is all about glycophorin. आपसे ये नहीं पूछेंगे कि वो ये रिसेप्टर इससे इंटरेक्ट होता है, ये इससे होता है, ये कोई नहीं पूछेगा. What they're gonna ask is using what the malaria parasite is infecting or invading the erythrocyte, human erythrocyte. So what will be the answer? Glycophorins. बस इतना याद रखना है, ठीक है? So I don't want just for you guys to remember. I want that जो उसके पीछे की स्टोरी है, why it is glycophorin, that also you should know. Okay? Otherwise पढ़ने में फिर मजा नहीं है. Okay. Now let's talk about the lipid bilayer and its types. So we have discussed what lipid bilayer is. Now we need to talk about the micelles formation and the other stuffs. Like why it is important? Because these micelles formation are there, which is helping in the drug targeting also. क्योंकि ये सारी चीजें दोबारा आएंगी when we will be discussing about the cancer and how the drugs are targeted to the cell. Like अब हमें टारगेट बोल रहे हैं तो इसका मतलब क्या है? इट हैज टू बी स्पेसिफिक टू दैट पर्टिकुलर सेल. ऐसा नहीं कि मैंने अगर ये ड्रग दे दिया तो वो नॉर्मल सेल को जाके मार दे. आई डोंट वांट दैट, राइट? So all these things will be discussed later, but for that we need to understand the chemistry behind that. Okay. So these are nothing but the amphipathic compound which has got both hydrophilic हेड, ये आपका हाइड्रोफिलिक हो गया जहां पे आपका फॉस्फेट ग्रुप प्रेजेंट रहता है, ठीक है? एंड द हाइड्रोफोबिक टेल, दैट इज योर कार्बन चेन इज देयर. They interact with the aqueous system in vitro to form a sphere. So this you know, right? In order to avoid maximum contact with the water, what they do, they form a sphere. Now micelles have only one polar surface, which is side presented to the aqueous phase. Clear? Now there is something called liposomes also. I think it will be there. Okay, so before we proceed, I'll just explain. So micelles क्या हो गया आपके? Single layer. Okay, जैसे आपका माइसिल ऐसे फॉर्म होगा. Sorry, this is wrong. ऐसे होगा. So where it will be like this. Polar head बाहर की तरफ, hydrophobic inside. Now, what are liposomes? Have you guys heard about liposomes? Because liposomes are things which are used for drug targeting. Now, liposomes, I hope I have the structure because I'm not that good in drawing. So, liposomes are like this. This is your one layer of plasma membrane. This is another layer of plasma membrane. Okay? ये कैसे होता है? ऐसे. Now, because of this, what will happen? They are going to circularize like this. फिर एक और लेयर आ जाएगा, जिसकी वजह से क्या होगा? यहाँ पे कैविटी है. There is nothing inside. Okay, this is aqueous. This is your membrane, and here it is cavity. Now, this cavity you are going to put your drug here. And then you use this as the capsule में डाल के in the powdered form, and whenever any membrane is coming, so this membrane composition and your cell membrane compositions are same, so they can fuse with each other and drug can go inside the cell. इतना सा समझ में आया क्या? To everyone, is it clear?
Yes, ma'am.
Yes.
Yeah, okay. Just understand this concept, like why liposomes are used, okay? Because of the similarity in the membrane composition. So जो भी आपका cell रहेगा, let us say this is your cancer cell. Since this membrane and this membrane are having the same composition or similar, I shouldn't say same, but similar. And so what will happen? This phospholipids will fuse and the drug which is there can be go inside. ठीक है? And cancer cell में cytoplasm जो रहता है, the pH is different. And that see, if anything comes inside, what happens? Directly it goes to your lysosomes, क्योंकि उसको नहीं पता क्या आया है बॉडी के अंदर, right? So it is going to the lysosomes for the check. Lysosome में pH आपका कम रहता है, if you remember, it is around 3 to 5. Okay, it is acidic pH. That acidic pH is going to make your drug active, and that active drug is finally going to kill your cancer cell. कुछ भी signaling blockade कर देगा, and it will cause your cancer cell to undergo apoptosis. This is the entire scenario by which maximum of your cancer drugs are working. We will discuss in detail. Don't worry about it. Okay.
Yes, ma'am.
Okay. So now it's like what are the different types of missile formation? Okay. So normally oil in water हमने देखा ही है. Okay. This is same thing like what we put a small drop in the water. How do we see a small spherical formed we see, right? सेम चीज अगर ऑयल है बहुत सारा उसमें अगर आप पानी डालोगे तो क्या होगा एक छोटा सा ड्रॉप बन जाएगा एंड इट विल टेक द शेप ऑफ द स्फीयर. नाउ व्हाई वी ऑलवेज गो विद द स्फेरिकल शेप? क्यों वो रेक्टेंगल या कोन नहीं बनता है? व्हाई इट इज ऑलवेज स्फेरिकल शेप the droplets? What is the reason or the physics behind it?
Sphere takes the least amount of area, amount of volume.
Is it a surface area or volume? What is it?
Volume. It takes the least amount of volume.
Nah, it is surface area only. It takes the less amount of surface area. Okay? Yeah. Okay. Good. So now oil in water. Oh, one second. So now coming to the oil in water, you can see again हाइड्रोफिलिक हेड बाहर रहेगा, फोबिक आपका इनसाइड रहेगा. ठीक है. नाउ same way if I say water in oil, it is going to be the opposite. So the polar head will be in the inside face facing the fatty acid tails outside, right? Just uska ulta hoga. Now the structure of micelles is very stable because of hydrophobic interactions between the hydrocarbon chains and the attraction of polar head group to water. This is the reason why micelles are very stable. Okay, they're important in intestinal digestion and absorption of lipid also. So this, when we talk about like animal physiology mein, how micelles are actually helping, especially जब आप trans fat खाते हो या ghee rich food you eat, wherever oil is there. So how these micelles are actually helping in the digestion. ठीक है. Like lipid absorb करता है, it takes to the adipose tissue and helps in dumping over there. It helps in making us fat. Okay. Now, mobility of lipid components in the membrane. तो ये भी हमने पहले discuss कर चुका है. This you already know actually. ठीक है. Where in which we have talked about the flippase, floppase, and scramblase, right? So this is the same thing. So we know that the phospholipids are not static in nature, they are dynamic. So they can rotate freely around their long axis, diffuse laterally within each leaflet. इसका क्या मतलब? इनर से आउटर में जा सकता है, आउटर में इनर जा सकता है. ठीक है. एंड दे कैन रोटेट देयर इटसेल्फ. एक अगर ऐसा फॉस्फोलिपिड है तो दे कैन रोटेट स्टैंडिंग इन देयर ओन पोजीशन एंड दे कैन रोटेट ओवर देयर. नाउ ऑल फॉस्फोलिपिड्स ऑफ प्लाज्मा मेम्ब्रेन आर मोबाइल, दे आर नॉट फिक्स्ड, दे आर मूविंग लैटरली. द ट्रांजिशन ऑफ ए लिपिड मॉलिक्यूल फ्रॉम वन लीफलेट टू द अदर इज कॉल्ड ट्रांसवर्स डिफ्यूजन. अगर सिर्फ आउटर में डिफ्यूज हो रहा है, इट इज लैटरल डिफ्यूजन. इफ इट इज फ्रॉम आउटर टू इनर गोइंग और इनर टू आउटर इट इज गोइंग, देन इट इज ट्रांसवर्स डिफ्यूजन. सिंपल. दिस ऑलरेडी नो, वी ऑलरेडी नो, राइट? एंड इसको हेल्प करने वाला कौन है? हमारा फ्लिपेज, फ्लॉपेज एंड स्क्रैम्बलेज. ओके. Now something about the lipid rafts. This is I actually wanted to discuss. Okay, so lipid rafts मैंने पहले भी mention किया था, and here the cholesterol play very important role. Okay, so if you see in the figure also, you see that if this is the cholesterol, it is rich in sphingolipids and the cholesterol. Now, if you remember, I have said that the phospholipid composition in the outer and the inner, it is different. If you remember, right? Phosphatidylcholine, serine, ethanolamine, inositol, and sphingolipids were there. है ना? Now, tell me what is present on the outer leaflet of the plasma membrane and what is present on the inner leaflet of the plasma membrane. Who can tell me? Shruti? Okay, Tanvi, याद है? Akshaya?
No, ma'am.
Nahi? Okay, Akshaya?
Phosphatidylcholine, serine, and inositol are present inside. Rest are outside.
Wrong. Simple. Phosphatidylcholine and sphingolipids are outside, rest are inside. मैंने क्या बोला था आपका serine तो inside ही होना चाहिए because during apoptosis it gets exposed to outside, right? Simple था, सिर्फ PC आप याद रखते हैं, it is present in the outside. So phosphatidylcholine and sphingolipids are present on the outside, rest is present on the inner leaflet. Okay. Now look at over here. तो ये देख के पता चल जाता है स्पिंगोलिपिड्स आपका आउटसाइड में ही है एंड ऑफ कोर्स जीपीआई ग्लाइकोसिलेटेड फॉस्फोइनोसिटोल लिंक्ड प्रोटीन्स करेक्ट रिमेंबर सो यहाँ पे दिस इज रिक्वायर्ड फॉर योर सेल सिग्नलिंग मैंने पहले ही बोला आपको then cholesterol is there sphingolipids are there so all this together they form a raft okay They are the membrane microdomains enriched in sphingolipids, cholesterol, and certain lipid-linked proteins. ये आपको याद रखना है. This is really very important कि sphingolipids, cholesterol, and lipid anchor proteins or lipid-linked proteins are present. ठीक है? It's really very important. Now look at the figure over here. Let us say that this is your plasma membrane. This lavender color highlighted ones is your rafts. So what they want to show is that these particular areas are totally rich in cholesterol, sphingolipids, and the other glycolipids. Okay? Now, how do they move? That is also important. Look at this animation. So what do we see here? You can see that this is like something, you know, it's like over the top of the ocean. So everything is mobile. These phospholipids are also mobile. And onto this purple color thing is your lipid rafts, wherein which later you can see here, see certain integral proteins are present, right? So they carry the cell signaling molecules over here on the top of it. And this is how they always keep on moving. So जब भी कोई दूसरा cell आता है and the interactions or any kind of signaling is required, these rafts come together because they are providing the signaling receptors so that they can have the interaction. Clear? So this is just for your clear imagination, like how on the surface of the cell membrane these rafts are moving. Is it clear? Yes?
Yes, ma'am.
Okay. So now lipid rafts are there are two types are there, made up of caveolin and non-caveolar. So what are these? These are the proteins again. ठीक है? Now caveolae, non-caveolar मतलब वहाँ पे कोई protein की जरूरत नहीं है. Now in the case of the caveolae, they are also a subset of lipid rafts around 60 to 80 nanometer. Now they give plasma membrane invaginations. Can you see in the figure? So these proteins they come to the membrane and they invaginate. ऐसे बना देते हैं. Okay, can you see? This is the entire plasma membrane. So many invagination is there. So they are formed by the oligomers of the membrane protein caveolin, which interacts with the cytoplasmic protein caveolin. अब इसका क्या मतलब? See on the surface here. Okay, ये cytoplasmic region हुआ आपके plasma membrane का. So it has got a protein called caveolin. To which your caveolin as a dimer comes and binds and helps in the formation of the invagination. Okay? So caveolin interacts with cholesterol. A high content of membrane cholesterol is required for the formation of the caveolae. So what they form is the caveolae. They bind to the cytoplasmic protein caveolin, caveolin dimer, and results in the formation of a caveolae, or in simple words, membrane invagination. Okay? Especially they are seen in the case of the endothelial cells and the adipocytes to increase the surface area. Now, they are involved in variety of cellular activities like endocytosis, cell signaling, lipid transport, protection of the plasma membrane against mechanical stress, etc. Clear? Is clear? Okay. ये आपको याद करना ही है. This is really very precise slide I made depending on the exam जो भी पूछा गया था. ठीक है? Of course, this was not asked. This was asked, and of course the endothelial and the adipocytes. Now, coming to the raft associated protein. So this is the best example. Okay, how cholera toxin is causing or it is affecting the host cell. Now, it is the cholera toxin B subunit, which is released by the cholera. Okay. So this is actually a toxin. Now, how it is affecting us, that is what is shown in this figure. So it consists of the membrane binding domain of the cholera toxin CTXB, which binds to 5 copies of its lipid receptor on the plasma membrane of the host cell. Okay, so this multivalency of the binding gives the toxin the ability to reorganize underlying membrane structure by cross-linking otherwise small and transient lipid rafts. So what is happening is that, see, नॉर्मली आपके राफ्ट में हमारे रिसेप्टर्स ऐसे फ्लोट कर रहे हैं, ठीक है. दिस इज योर राफ्ट एंड यू कैन सी दिस GM1 PLAP receptors are there. Now once cholera toxin is released, okay, they go and bind to these receptors, and what is happening, they bring all the lipid rafts together. If you see, ये सारे को एक साथ merge कर देता है and makes it possible for the cholera toxin to get inside the cell of the host. तब क्या होगा, it is going to release another protein which is going to form the pore. यहाँ पे एक पोर बनाएगा. So पोर बनाएगा तो क्या होगा, it makes it hollow pore like this. ठीक है ना. Pore is, again, if you remember, it is nothing but the beta sheets or the alpha helices transmembrane proteins which allows the access of the outer to inner and inner to outer. तब क्या होगा, टॉक्सिन सीधा होस्ट सेल के अंदर जाएगा and it will start killing the host cell. ठीक है, clear? Now, why this I am explaining, because again this question has come, like how cholera toxin is interacting with the host cell. So it actually brings or cross-links all the lipid rafts of the host cell. Is this much is clear? कि जैसे जैसे हम what to say MCQs भी करेंगे, it will become more clear. Okay, so far so good, guys. Able to follow? Any doubts in today's lecture?
No, ma'am, it's clear.
Okay.